several things to deal with here. Number one, I have good news for you. That shut everybody up. The good news I have for you is on the final exam, I let you have a note card with notes that you fill out 100% by hand. No printed note cards, no figures pasted on. Everything on the note card has to be in your own handwriting. It's a big note card, but you have to get the note card from me. Okay? I will bring the note cards to class on Friday, and if you, for some reason, aren't here on Friday, you will have to come and get it from me later, after Friday. Okay? But I will hand out the note cards in class on Friday. All right? Everybody with me? You have to get the note card from me. And you have to have a note card. Meaning that when you come to the final exam, if you do not have a note card that you got from me, you will lose points. That's the bad news. But hey, if you get a note card, right, then you're all set. Everybody understand what I'm talking about? So you have to have a note card from me that you will bring to the final exam. All right? You can have a blank note card from me. I don't care, as long as you have a note card from me at the final exam. I'm not going to make you use a note card. I actually have a couple students a year that say, I don't want to use a note card. Well, okay, that's fine. But bring the note card that I give you to class. Does that make sense? Okay. So make sure you come to class on Friday. If you're, and don't come to me Friday at noon and say, oh, I can't be in class today. Can I get a note card? Because I will say no. You can come see me on Monday. Is that fair? I like this. Okay, fair. All right. All right. Well, in the meantime, we have a fair amount of material to cover, so I want to get through covering it. Let's try to do that. And last time when I was talking, I finished talking about the Z scheme. I didn't even give it that name, but the Z scheme of photosynthesis. And that was that up, down, up, down. That's why they call it the Z scheme of photosynthesis. Today, I want to cover some other aspects of photosynthesis, including the dark reactions, and then I'll start talking about the nitrogen fixation, which will be at the end, closer to the end of the lecture. Okay. Well, one of the things that happens with plants is that plants, maybe not in Oregon, but plants get a lot of sunlight. And because they get a lot of sunlight, especially in the summertime where the days are long, that they actually have a problem. What's the problem? The problem that they have is that just like you have with your mitochondria, they have to have, okay, an electron acceptor. Okay? They have to have an electron acceptor. Okay? The electron acceptor in normal light reactions of photosynthesis is actually NADPH. I'm sorry, NADP to make NADPH, right? Well, if you have a lot of sunlight and the cells aren't using that NADPH up very rapidly, then you've got no place to put electrons, but you still would like to really use all that energy that's coming down from that sun in that sunlight. So cells, plant cells actually have a backup mechanism that allow them to bypass that. It's called cyclic photophosphorylation. And it basically involves what you see on the screen here. This is photosystem 2, absorbing a photon, okay, passing it off to the carriers that do the proton pumping. Okay, so this is a different system than we saw before. It's passing electrons off to the carriers that do the proton pumping, and then that electron is leaking back to photosystem 2 again. So it's a cyclic system where that electron is just cycling between here. But the beauty of this system is because the electrons are going through these proton pumping systems, that protons are still being pumped. That turns out to be very useful because what does proton pumping do? Proton pumping allows synthesis of ATP. So even though this cell would be running out of electron acceptors, it still can make ATP. Right? We can't do that because when we run out of an electron acceptor, when we run out of oxygen, right, then no more protons get pumped. That's why we suffocate, that's why we die. These guys have a way around that, and they have that way around that because it allows them to make more ATP. That's a very important consideration. So cyclic photophosphorylation is an alternative mechanism for these cells to keep synthesizing ATP even though they have no electron acceptor in the form of NADP. Okay. Man, that's a lot of stuff. It's actually what I've already shown you. I'm not going to go through that again. Okay. Photosynthesis in an anaerobic bacterium is different. And what I will tell you is that the synthesis in an anaerobic bacterium is very much like what you see in photosystem 2 in that cyclic photophosphorylation that I showed you. Okay? They want to bypass an anaerobic bacterium, if it's an obligate anaerobe, wants to bypass the synthesis of oxygen because oxygen is actually poisonous to it. So if it's going through that photosystem 1 and splitting water, then what's happening is it's making something poisonous to itself if it's an obligate anaerobe. So as a consequence, anaerobic bacteria that are obligate anaerobes will primarily run photosystem 2 in that photocyclic mechanism that I just showed you. Okay. Now, that said, I finished the light reactions. We've covered all the different considerations that we're going to have with respect to what happens when the lights are on. I'm now going to turn our attention to the dark reactions because photosynthesis isn't over yet. Right? The photo part of it is over, but the synthesis part is just getting ready to start. Now, the dark reactions, as I said yesterday, do not require dark. The light reactions require light, but the dark reactions do not require dark. All right? Well, the dark reactions occur in a cycle that we call the Calvin cycle, named for the person who was instrumental in discovering the reactions of the cycle. I told you earlier that the pentose phosphate pathway, which is a pathway that I said was very complicated, is related to a pathway in photosynthesis, and this is the pathway that it's related to. We see many of the same intermediates that we saw in the pentose phosphate pathway now in the Calvin cycle. Okay? The good news is I'm not going to make you know those reactions, except for a couple of them. Okay? But I want to point out some of the things that are in here that are important to understand. Okay? This process starts with a molecule, and this has a starting point, by the way. It's different from pentose phosphate pathway. It has a starting point. It starts with a molecule that has a mouthful of a name. It's called ribulose 1,5-bisphosphate. You can call it RI-15BP. The book calls it RU, but I call it RI-15BP. Okay? This is a 5-carbon molecule, and this 5-carbon molecule is a substrate for an enzyme called ribulose bisphosphate carboxylase. That's a mouthful of a name. Most people call that enzyme RuBisco. That's what you heard in the song yesterday. RuBisco, R-U-B-I-S-C-O. Ribulose 1,5-bisphosphate is a substrate for RuBisco. And there's a second substrate. The second substrate is the molecule carbon dioxide. You notice this is a carboxylase, and a carboxylase will put carbon dioxide, put a carboxyl group onto something, and that's what this is doing. We know that plants generate oxygen. Another thing that plants do is they assimilate carbon dioxide. They take carbon dioxide out of the atmosphere and they build it ultimately into glucose. So what the Calvin cycle is all about is what I just told you. Taking carbon dioxide out of the atmosphere and using it to make, ultimately, glucose. Well, this happens, please turn that off. This happens 1 carbon at a time. Okay? We see the carbon dioxide coming in here. We see all these reactions, and ultimately, we see glucose being produced. Now, the upshot of this is we can't produce glucose from 1 carbon. we have to produce glucose from 6 carbons. So it takes 6 of these and 6 of these ultimately to make 1 glucose. One of the things that people get confused about when they study the Calvin cycle is, oh my God, it's 6 of these, 12 of these, 3 of these, 4 of these, 1 of these, 2 of these, 6 of these, and finally 1 of glucose. We're not going to worry about that. Okay? We're not going to worry about that. The numbers aren't going to matter to us, although I do think you should know it takes 6 carbon dioxides and 6 ribulose 1,5-bisphosphates to ultimately make 1 glucose. Right? So the process I've just described to you is called carbon fixation. Carbon dioxide is being taken from the atmosphere and being made into glucose. Because of this reaction, essentially everything that we know of on Earth occurs as far as living systems go. Because we're using carbon dioxide and we're making an energy source. The energy source in this case is glucose. The plants use it for energy. The things that eat plants use it for energy. And ultimately, essentially all of biological energy is coming from this reaction. RuBisco is described as the most abundant enzyme on the face of the earth, because every plant has it. Okay? As we will see, RuBisco is an odd enzyme in that it's very inefficient. This always disturbs people. It's inefficient. Nature is not perfect, but nature is as perfect as it needs to be. Okay? It's like when you're a kid, right? You're not perfect, but you're good enough that your parents don't kill you. Right? Just good enough to get along, good enough not to get in trouble, right? But not perfect. What's the fun in being perfect? Well, it's the same way with biological systems. Okay, so there are all kinds of intermediates in here, all right? No, I'm not going to ask you to know them, but I'm going to point some of them out to you, okay? You see within here glucose 6-phosphate. You see fructose bisphosphate. You see fructose 6-phosphate. You see 3-phosphoglycerate, okay? You see a lot of things that you saw in glycolysis. And some of these enzymes overlap with glycolysis. And that turns out to be rather convenient. Okay? These are common things that are found in cells, and these are common things that are common also to the pentose phosphate pathway, as I mentioned before. Okay. Here's that carbon dioxide, or that carbon fixation reaction. Ribulose 1,5-bisphosphate is gaining a carbon dioxide to make an intermediate that's unstable, and that unstable intermediate falls apart and makes 2 molecules, and this is another one you should know, of 3-phosphoglycerate. So we're starting with a 5-carbon molecule, adding a 1-carbon molecule, and ending up with 2 3-carbon molecules. 3-phosphoglycerate, as you know, is an intermediate in glycolysis. As well as intermediate in gluconeogenesis. I started to say gluconeogenesis. And the reactions of gluconeogenesis will take us from 3-phosphoglycerate ultimately back up to glucose. The rest of it that you saw on that screen is balancing the equation. Okay? That's really what's happening here. So this is not a complicated process, even though it looks like a complicated process. Okay. This is what the enzyme looks like. Draw that on the final exam. Okay? And art will count on the final exam. Right? The closer you are to the accurate structure of that enzyme, the better. So bring some colored pens. All right. And there's more about the enzyme. Now, this enzyme turns out, as I said, not to be very efficient. Not to be very efficient. And people don't really understand why it's not more efficient. People would like to make this enzyme be more efficient. And perhaps one of the reasons it's not very efficient is that there's a side reaction that can occur that this enzyme catalyzes, and that side reaction is shown right here. Okay? Ribulose 1,5-bisphosphate, in addition to combining with carbon dioxide via RuBisco, can also combine with oxygen via RuBisco. And when that happens, we don't get 2 molecules of 3-phosphoglycerate because that would mean we had 6 carbons. We don't have 6 carbons here. We have 5 carbons here plus molecular oxygen. If molecular oxygen is used by the enzyme, then what happens is we get 1 molecule of 3-phosphoglycerate as we expect, and we get a molecule of phosphoglycerate, all right, which falls apart and makes glycolate, which ultimately makes glyoxylate. Anybody remember the glyoxylate cycle? Where did the glyoxylate cycle occur? Plants and bacteria. Glyoxylate turns out to be a reasonably toxic molecule. Good exam question might be, why do you suppose plants have a glyoxylate cycle? To take care of that molecule right there. They're also capturing energy. Because this 5-carbon molecule, if they just threw this glyoxylate away, would be losing energy for the plant when this reaction occurred. This helps to keep that energy that's in this molecule from being lost. The glyoxylate cycle is very beneficial to plants. Clear as mud? Questions? Here's where I got to pause. Yes, there's a question. I'm sorry? Which molecule did glyoxylate what? So the glyoxylate cycle is using the glyoxylate that's produced here, okay, so that it doesn't go to waste. So the glyoxylate cycle, if you recall, takes glyoxylate and ultimately makes oxaloacetate. Okay? Other thoughts? Questions? I'm trying to observe the 30-second rule here. If I'd done that earlier in the term, we'd probably be way behind, so maybe it's a good thing I didn't do that. How frequent is it taking up O2 versus CO2? It's reasonably frequent. It's reasonably frequent. That's one of the reasons that the enzyme is not very efficient. Yeah. Is this a way of making oxaloacetate? Is that your question? So cells in general, his question was, would the plant have enough oxaloacetate if this reaction didn't occur? I think that's the question, right? Yeah, cells in general have pretty abundant sources of oxaloacetate because, for example, they can always convert aspartic acid into oxaloacetate. So I wouldn't say there's a shortage of oxaloacetate, no. That's not the reason this reaction is here. But this inefficient reaction is happening in the vast majority of plants on the face of the earth. So there are obviously advantages to having this reaction occurring. Okay. Well, the last thing I want to say about the dark reactions relate to what are called C4 plants. Okay? How many people have heard of C4 plants? What do you hear about C4 plants? What do you learn about them? Can't remember. Okay, very honest person right here. Anybody have a thought about C4 plants? What's that? Cactuses, yeah. So C4 plants, the reason people talk about C4 plants is they're fairly efficient at what they do, and they're fairly efficient at not losing water. We see them in things like cactuses and so forth, where water is a limiting reagent. Okay? You're going to see why that's the case here. All right? If we were to study the reaction I just described to you, that is the RuBisco reaction occurring in a regular plant, it would be occurring on the cells of the surface of the plant leaf. And the problem with that is, if the RuBisco reaction were occurring out here, this plant leaf is exposed to a lot of atmospheric conditions where if the humidity is low, for example, a lot of water is going to be lost through that outer cell. Okay? So plants, C4 plants, have evolved a way that reduces the need to expose as much of themselves as possible to water. All right? How do they do that? Well, what they do is they've got a shuttle system that grabs carbon dioxide, and instead of letting that reaction occur out here, the reaction itself is occurring further inside of the plant, where there's more water and more water needs. Okay? Out here in these surface cells of a C4 plant, and yes, we're looking at a C4 plant, the carbon dioxide is captured, but the reactions that the Calvin cycle occur in are not on the surface of the plant leaf, but instead internal to it. So this shuttle system allows the plant to move things inside. Now, the bottom line of a C4 plant is that the RuBisco reaction, the Calvin cycle, all are occurring just like they're occurring in a C3 plant. The other plants are called C3 plants. Okay? I'll tell you why in a second they're called C4 plants. But in a C3 plant, which is where the RuBisco breaks down into 2 molecules of 3-phosphoglycerate, right? The same reactions are occurring that are occurring in the C4 plant. That's very important to understand. It's not different. The reason we call these C4 plants is because the first molecule that's made does not have 3 carbons. The first molecule that's made has 4 carbons. That's the 4 part of a C4. In the C3 plant, we saw RuBisco plus, I'm sorry, not RuBisco, ribulose 1,5-bisphosphate plus carbon dioxide gave 2 molecules of 3-phosphoglycerate. Those were 3 carbon molecules. Okay? The first reaction here has a phosphoenol pyruvate, which has 3 carbons, grabbing a carbon dioxide to make oxaloacetate, which has 4 carbons. There's your 4-carbon molecule. Oxaloacetate gets reduced to malate, and then malate is moved internal to where the Calvin cycle reactions are going on. What does malate do? Well, it dumps off a carbon dioxide, and it dumps it off onto a ribulose 1,5-bisphosphate. So all that's going on out here is a system for grabbing carbon dioxide and bringing it internal to the plant. That's all that's going on here. It starts out as a 4-carbon molecule, but in here we still have the RuBisco reaction going on. The RuBisco reaction is still occurring internal in that plant, right here. Then all those other reactions that I described to you for making glucose, and by the way, there's glucose being made right there, all those reactions that you saw in the C3 plant are still going on and still producing glucose. The difference is that the first thing that's made has 4 carbons instead of 3. But we're still making 2 molecules of 3-phosphoglycerate down here. Does that make sense? Gesundheit. It's the most unusual sneeze I've heard. Nobody's saying anything. If you don't say anything, I will move on. Because that's what I do. Yes? Is what? Is the Calvin cycle the same as the citric acid cycle? No, the Calvin cycle is related to the pentose phosphate pathway. Okay? So though it looks like it's a circle, it's not really a circle. All right? Although there are components of it that do come around cyclically. But it's not the citric acid cycle, no. Am I going to ask you to draw this on the final? No, I'm not. All right? But you should know why it's a C4. You should know how a C4 is different from a C3. But you're not going to draw the reactions. In general, you've seen I haven't made you draw any reactions. Okay? All right. That's the last of what I want to say about photosynthesis. And that was... Nah, you don't want that. Okay. Well, let's turn our attention now to finish out the term on the topic of nitrogen metabolism. We're almost there. The end is in sight. Okay? Nitrogen metabolism is a topic in biochemistry that is not the most glamorous one that's out there. But for a nutritionist, it actually is a pretty important set of considerations. The nitrogen cycle, or the nitrogen metabolism, it's often called the nitrogen cycle, is something that is not universal to an organism. And if we think about this more in terms of nitrogen balance, we'll be better off. We have to balance within our bodies the proper amount of nitrogen. Too much nitrogen is not good. Too little nitrogen is not good. And nitrogen is important because, remember, amino acids are important, and nitrogen is a component of amino acids. Nitrogen is also a component of nucleotides. The problem that we have as animals, as most organisms on the face of the earth have, is we cannot use nitrogen from the atmosphere. We cannot, underline that, we cannot use nitrogen from the atmosphere. Okay? Well, how does nitrogen get into living systems? Okay? Well, there are organisms called nitrogen-fixing bacteria that are responsible for essentially all of the biological nitrogen on the face of the earth. Nitrogen fixation, you've heard of that, right? If we were to lose all of the nitrogen-fixing bacteria on the face of the earth, folks, we would be in deep doo-doo. There's a scary thought. We would be in very deep doo-doo. When you hear people worrying about water pollution and soil pollution, you think about these bacteria that we're dependent upon are living in these environments, we think, wow, we got to be careful. Because without that, there are not going to be any amino acids. And I hate to tell you, but you are rather dependent upon amino acids. Okay. Well, the nitrogen cycle is depicted schematically here. And the nitrogen cycle is more a cycle of what's going on the face of the earth and even under the face of the earth. Okay? Nitrogen makes it into the biosphere, as I said, coming from bacteria, and most commonly those bacteria are feeding plants. The plants are taking the nitrogen from the bacteria, and there are nitrogen-fixing plants. They're called nitrogen-fixing plants, but it's actually the bacteria in the roots of the plants that are making that possible. Okay? Legumes, L-E-G-U-M-E, are plants that contain nitrogen-fixing bacteria in their roots. Okay? Nitrogen-fixing bacteria can, in fact, take nitrogen from the atmosphere and make it biologically available. They have to alter it for that to happen, but they're making it biologically available by a series of different reactions, some of which I will show you. Okay? Well, they make it into plants. Animals eat plants. There's a bunny rabbit eating a plant. Bunny rabbits poop. And poop, as everybody knows, is good fertilizer. And the reason poop is good fertilizer is because it's full of nitrogen. Right? So we've already gone through a simple part of the cycle doing that. We eat bunny rabbits. It's kind of disgusting, but it's true. People eat bunny rabbits, right? And so animals eat animals, and that's another way that they get their nitrogen. Okay. Well, first I want to talk about one of the ways in which nitrogen is fixed. When I say fixed, I'm basically talking about taking it out of the atmosphere and putting it into a form that's biologically useful. All right? This is a very energetically expensive set of reactions that go on. Very energetically expensive set of reactions. Okay? Here's molecular nitrogen. It takes 6 electrons to reduce molecular nitrogen into 2 molecules of ammonium ion. Where I say ammonium ion, you can think of ammonia. Because if you put ammonia into water, you get ammonium ion. Okay? There are a series of reactions that make this all happen. Okay? A series of reactions that make this happen. But ultimately, the bottom line is that it takes electrons to make this happen, it takes protons to make this happen, and look how much ATP it takes to make this happen. 16 ATPs to fix 2 atoms of nitrogen. Okay? That's a lot. Okay? It's not surprising that the bacteria that do this live in a symbiotic relationship with plants, because the plants can provide the energy necessary for the bacterium or the bacteria to make these reactions go, and in exchange the bacteria are giving up the reduced nitrogen for the plants to use. Okay. This is a very complicated set of reactions. Now what I've just done is I've just skipped over a ton of things relating to nitrogen metabolism. Why did I do that? Well, nitrogen metabolism, nitrogen fixation, is actually not just the one reaction I showed you on the screen, but there are other reactions and other considerations for how atmospheric nitrogen can be used. What I want to do instead, since we don't have the time to go through and talk about all those types of reactions, what I want to do instead is focus more on what's happening in our bodies, because I think that's really where we're interested in nitrogen metabolism. Okay? When we look at nitrogen metabolism in our body, what we discover is that nitrogen moves from nitrogen donors to nitrogen acceptors. And specifically, it's moving in our body as amines. That's reduced nitrogen, NH2s. Okay? One of the primary sources of nitrogen, and actually two of the primary sources, are glutamate, that's glutamic acid, and glutamine. Okay? How do I go from a glutamate to a glutamine? Well, there are many ways that that can happen, but the one you see on the screen takes ammonium ion, it takes ATP, it goes on to glutamate, and when I do that, I've got glutamine. Now, you probably don't remember the structures of all these, but I will tell you that glutamate has 1 reduced nitrogen on it. Glutamine has 2. So this guy has gained a reduced nitrogen. And if I take one of those reduced nitrogens off of glutamine, I'm going to go backwards and make glutamate. Okay? What's this complicated figure showing me? This complicated figure is showing me that there are several things that will inhibit this reaction from occurring. In fact, there's a large number of things that will inhibit this reaction from occurring. Glycine, alanine, carbamyl phosphate, AMP, CTP, glucosamine 6-phosphate, tryptophan, histidine, serine. Wow. Do I have to remember all those? No. All right. What's inhibiting this reaction in general from occurring is the fact that you have abundant molecules with nitrogen already in them. I have abundance. I don't need to be moving more nitrogen. Let's do something else with this. Okay. Now, glutamine can be used to make each of these molecules that you see there, because each of these has a nitrogen in it. So one of those amines that are in glutamine can be used to donate and make each one of these molecules. So each of these molecules are made from glutamine. So if you make too much of them, the message is quit making more glutamine because we don't need any more of these molecules. It's a feedback type inhibition. Okay? Now, this is a set of reactions that I will expect you to know something about because they're fundamental to understanding the movement of nitrogen between molecules inside of cells. Okay? These reactions are called transamination reactions. Transamination reactions. And the enzyme that catalyzes them is known as a transaminase. Okay? I have to back up. I apologize. This is actually showing you the one way that ammonium ion can be taken up. Okay. This reaction at the bottom is the one you already saw. Ammonium plus glutamate gives glutamine. We can similarly put an ammonium onto alpha-ketoglutarate and make glutamate. I apologize. These are not the transamination reactions. Okay. But I will be getting them. Don't worry. The difference between, where do we see alpha-ketoglutarate? Citric acid cycle, right? The difference between alpha-ketoglutarate and glutamic acid is an amine. There's the amine, there's an oxygen. And the reason I started to call this a transamination is what we will see in the transamination reactions is there's swaps of oxygens and nitrogens. Okay? The difference between glutamate and glutamine is, again, a nitrogen. There's an extra oxygen on here. That oxygen is replaced by an amine over here. Okay? In both cases, ammonium ion is being taken up and used to make an amino acid that has another amine in it. Okay. Let's see. Where's my transaminations? Here we go. Okay. This was what I started to tell you, and you'll see why I got confused myself. All right. Transamination reactions, catalyzed by the enzyme transaminase, as I noted, and they involve swaps of nitrogens and oxygens between different molecules. Okay? It takes two sets of molecules for transamination reactions to occur. You have to have a nitrogen source, and you have to have an oxygen source. Okay? So here's one set of reactions. Here's glutamic acid, and you can see the amine that's involved in this swap right here. Okay? Here is an alpha-keto acid. That could be alpha-ketoglutarate. That could be pyruvate. That could be oxaloacetate. They're all alpha-keto acids. The alpha-keto acids are the source of the oxygen that's used in the swap. What the transaminase is doing is it's swapping the oxygen and the nitrogen. When I do that, if I take the nitrogen off of glutamate and replace it with an oxygen, I get alpha-ketoglutarate. And I get an alpha amino acid because where that oxygen was, now there's an amine, and that is an alpha-keto acid. Same thing down here. Here's glutamate. Here's a specific example. Glutamate plus oxaloacetate. Glutamate's the source of the amine. Oxaloacetate's the source of the oxygen. I end up with alpha-ketoglutarate, and I end up with aspartic acid. Right? These guys have long names. Pyridoxal phosphate-dependent aminotransferase. Glutamate aspartate aminotransferase. We will call these enzymes by one name, transaminase, as I said. Because all of these enzymes work by the same general mechanism. Same general mechanism, transaminases. Okay? They're swapping amines and oxygens. Well, what's happening here is nitrogen is now moving from molecule to molecule to molecule. Does this reaction go to the left or to the right? Well, it depends. Does the cell need glutamate, or does the cell need aspartate? If the cell has an abundance of glutamate, and it doesn't have as much aspartate, that reaction is going to run to the right. What if I put in pyruvate here instead of oxaloacetate? If I did that, pyruvate looks just like this, except for it would cut off this top carboxyl group right here. The bottom 3 carbons are pyruvate. If I did that same reaction, I would end up with alpha-ketoglutarate plus alanine. So if I need alanine, bang, I've got an easy way of making alanine. Or if I have too much alanine and I need some glutamate, I've got an easy way of making glutamate. So these transamination reactions are very good for helping the cell to balance how much of each amino acid that it has based on how much it needs. Very easy types of reactions to occur. These type of reactions we find abundantly with amino acids. Not all amino acids, but we find this abundantly with amino acids. Now, what I started to show you was this. When we look at amino acids and their synthesis, oh boy, this gets complicated, we talk about families of them. There are families of amino acids, all right? Alpha-ketoglutarate, you've already seen, can readily be made into glutamate. And glutamate can be made into these 3 amino acids. And I'm not going to make you know those. So therefore, everybody's going to stop writing at that point, right? We're not going to memorize that. But you should know that there's a family of amino acids that's made from glutamate. Okay? The amino acids are not nice, and amino acid metabolism is not nice and tidy like the citric acid cycle. The citric acid cycle, we've got boing, 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 and we're right back where we started. We just go around that cycle. Okay? We've got 20 different amino acids, and each one of those has to be made in a specific way. You need to know that we don't make in our bodies all 20 of the amino acids. The reactions that I'm showing you that are occurring are not all ones that are occurring in our bodies. They're occurring in other cells in some cases. The cells of the organisms that we are eating. So we make in our bodies about 10 of the 20 amino acids. We make about 10 of the 20, depending on how you count them. Okay? The other 10 we have to get in our diet, and anything we have to get in our diet, I'm sure you've gathered by now, is called essential. So about 10 amino acids are essential amino acids, and 10 we can make. Okay? There's an aspartate family. Same story. I'm not going to make you memorize that. Aspartate, again, related to oxaloacetate. Difference of a transamination reaction. A serine family. And you don't need to know this, but serine can be readily made from 3-phosphoglycerate. And 3-phosphoglycerate is found in glycolysis. It's found in the Calvin cycle. It's found in the pentose phosphate pathway. There's an abundance of 3-phosphoglycerate in cells. The pyruvate family. Pyruvate, I've already described to you, can go directly to alanine by a transamination reaction. Can also go to valine and leucine. Again, I don't care if you know which ones. Obviously, I would hope you know the pyruvate family can be made into alanine because that's a transamination reaction. The aromatics turn out to be fairly complicated. Okay? So we sort of group them all together in an aromatic family. They use some intermediates that we have to go a long ways from here down to making these compounds. The aromatics are much more involved. Their synthesis is much more involved than the others are. Okay? And finally, we have the histidine family, which is a rather large family, as you can see. But notice what it's made from. It's made from, what do we make from ribose 5-phosphate? Anybody remember? Nobody remembers? What's that? Well, it's found in the pentose phosphate. That's how it's made. But what do we make from ribose 5-phosphate? Why is that important? Nucleic acid. We make nucleotides from this. So nucleotides are a product of both ribose 5-phosphate and histidine. No, you don't need to know that. I'm just throwing that out for you. Okay, so we've got several families. All right. I think that you should know, I'm going to tell you what I think you should know. I think you should know all of the ones I've talked about with respect to transamination. I'll repeat them for you here. That includes oxaloacetate going to aspartate. That includes glutamate going to glutamine. That also includes alpha-ketoglutarate going to glutamate. So we see glutamate sort of central. Alpha-ketoglutarate to glutamate, glutamate to glutamine. And the third one I think you should know is pyruvate going to alanine. Now what you will find is because it's so easy to make aspartate, and it's so easy to make glutamate, and it's so easy to make alanine, that they're not very essential. We can make them pretty readily. Okay? Others are more difficult for us to make, and they are essential amino acids. Okay. The last thing I'll show you is just this figure here. And you'll notice I'm not asking you for a lot of specifics, and I'm not going to ask you for specifics here. But I show you this figure to show you, first of all, the complexity of amino acid metabolism. Okay? On this side, we're looking at the breakdown of amino acids. On this side, we're looking at the synthesis of amino acids. There's nothing here for you to memorize. This is just for you to get a perspective, okay? These amino acids are broken down into alpha-ketoglutarate, which can be oxidized in the citric acid cycle. These guys can be broken down into succinyl-CoA and oxidized in the citric acid cycle. These guys can be oxidized to fumarate and be oxidized in the citric acid cycle. Okay? Here are ones that go to acetyl-CoA. Here are ones that go to acetoacetyl-CoA. Anybody remember what acetoacetyl-CoA is? It's sort of a trick question. I mentioned it once and I said you didn't need to know what it is. And so you don't need to know what it is. But does anybody remember hearing it before? Yes, what was it? You're close, okay? It does relate to fatty acid metabolism. And what it relates to is the synthesis of ketone bodies. That's involved in the synthesis of ketone bodies. But you don't need to know that. All right, everybody's tired. I'm tired. Let's sing a song and call it a day. What do you say? This is a song I wrote specifically for the BB350 class a couple of years ago. And it's called the Anthem for BB350. Oh, the students taking BB350 we have an awful lot of things that we must know. With acetic acid buffer, Kevin Ahern makes us suffer. The exams could not be tougher. 350. There's amino acid side chains to recall. And the things it takes to make cholesterol. Anabolic, catabolic, Kevin Ahern's diabolic. I'm becoming alcoholic. 350. Everybody always laughs at that one. There must be a way to jam into my head all the metabolic enzyme names I dread. Can you help me learn the spaces where the endonucleases cut the DNA in places? 350. Oh, I must find a way to make a better grade, or my GPA will truly get waylaid. I can overcome frustration to achieve my aspiration on the last examination. 350. Here's the plan I made to help me to succeed. Fill the note card with the knowledge I will need. I put all of Ahern's quotes along with what each one denotes onto a massive stack of notes for 350. So there's just one teensy problem I must fix. It requires some very skillful penman tricks. Squeezing info I must store onto the card he gave before will mean a font the size of 0.14. Oh, the students taking BB350 have an awful lot of things that we must know. With acetic acid buffer, Kevin Ahern makes us suffer. The exams could not be tougher. 350. All right, guys. See you on Friday for... Oh, by the way, I have a surprise for you on Friday.